Hello guys and welcome back to Excelsior Academy. So today we are going to do lecture 7. In this lecture we are going to talk about the PI3 kinase or the AKT pathway. So previously we talked about the MAP kinase pathway. Now this is the another pathway. So slowly all the pathways which are involved in cell signaling we are going to complete. Okay. So this is the second major pathway after the MAP kinase pathway for the intracellular signaling downstream of the tyrosine kinases, and again they are stimulated by the G proteins. Okay, so now you can see that how important these G proteins are. And again, it is based on the secondary messenger, which is derived from the membrane phospholipid phosphatidyl inositol 4,5 bisphosphate, that is PIP2. So from here onwards, we will say PIP2 only. Okay, so it's a component of the plasma membrane which is localized to the inner leaflet of the phospholipid bilayer. So guys, if possible, try to understand what is happening is that if suppose this is your plasma membrane, outer and inner, to this inner leaflet, this PIP2 is actually attached. Okay. Now then it is phosphorylated on the third position of the inositol by the enzyme phosphatidyl inoside PI3 kinase. इसका मतलब क्या हुआ? So this kinase has added additional phosphate group to this PIP2, which is making them into PIP3. Okay. Now this is ready for the binding. Now what kind of binding is happening in order to carry out the signaling? This is what we are going to learn in this lecture. So the form of PI3 kinase, which is activated by the tyrosine kinase, again it has got the SH2 domain. Now, and it is activated by association with the receptor tyrosine kinase. So there's an alternative isoform of PI3 kinase, which is activated by the G proteins. So PIP2 gives you PIP3, which is critical for the cell signaling and survival. Now, the target of PIP3 is the AKT. ठीक है, सो PIP3 में जो बाउंड होगा फाइनली क्या करेगा, वो AKT को एक्टिवेट करेगा. Now PIP3 binds to domain of AKT known as the plextrin homology or the PH domain. So guys, अब तक हमने क्या पढ़ा, that we are going to talk about the PI3 kinase pathway, and इसमें from PIP2 it is getting converted to PIP3 by the help of the kinase. ठीक है, and then finally it is going to activate the AKT by binding to the plextrin homology. Okay, this will be very clear once you see the figure also, so don't worry about it. So I'll show you the figure. Now the interaction recruits AKT to the inner face of the plasma membrane, where it is phosphorylated and activated by the other two protein kinases. So those are your PDK1 and mTORC2, again which has got the pH domains. Now this pH domain is important because using this pH domain only it is able to bind to PIP3. Now the formation of the PIP3 and its association with PDK1 and mTORC2 as well as AKT with the plasma membrane leading to phosphorylation and activation of AKT. तो हुआ क्या है अभी तक, PIP3 will actually associate with mTORC2 and PDK1, finally activating the AKT. और जब AKT एक बार activate हो जाएगा, so what this AKT is going to do, it is phosphorylating the other target proteins, for example BAD, which is responsible for the cell cyber or the other transcription factors. तो जैसे हमने मैप काइनेज में भी देखा कि आपका जी प्रोटीन एक्टिवेट होता है, फिर वो RAS एक्टिवेट करता है, फिर वो RAF करता है, MEG, ERK, and then finally ERK is activating the other target proteins of the transcription factors. In the same way, in this pathway also, your PIP3 is binding to the PDK1 and mTOR, finally activating AKT. AKT is later on phosphorylating the target proteins and activating the transcription factors. ठीक है, तो सारे pathway same है, it's just that you need to remember the different proteins. So this is the pathway PI3 kinase or AKT pathway. So whatever we have discussed till now has been summarized over here. So let's check out the figure over here. So as I always said, it's always better to study the concepts with the help of the diagram. So let's check out the diagram over here. Okay, so first we have got the growth factor which comes and binds to the receptor, and these are nothing but your tyrosine receptors. So they will dimerize. So once they dimerize, there will be a cross phosphorylation and they will get the phosphorylated. So once they get the phosphorylated, it actually initiates the binding site. Okay, it forms the binding site which has got the SH2 domain. So your PI3 kinase has got the SH2 domain जो यहाँ पे आके bind करेगा. ठीक है. Now this PI3 kinase का क्या काम है? PIP2 को PIP3 में convert करना. So that is again with the help of ATP, it is adding the phosphate group. ठीक है. So PIP2 जो आपका inner leaf rate of plasma membrane में था, वो अभी आपका PIP3 हो गया. Now once this PIP3 is there, to this your PDK1 and mTOR C2 will come and get activate. They will bind. So अभी आप देख सकते हैं क्या हो रहा है यहाँ पे. पहले mTOR आया, mTOR C2 आया. ठीक है. And then you have got the PDK1 also. So they all come together and what happens is that they are going to activate your AKT. अब AKT को कैसे activate किया इन्होंने? By phosphorylating. ठीक है. So once this gets phosphorylated, it is going to initiate or phosphorylate the other proteins which is required for the downstream signaling. ठीक है. So you can go through this what we have discussed. Same thing is given over here also. So the another important point was given over here is that the AKT then phosphorylates a number of target proteins like BAT, transcription factors of the other kinases, including GSK3, which is inhibited by AKT phosphorylation. So GSK3 यहाँ पे आपका inhibit हो रहा है. ठीक है. So because GSK phosphorylates metabolic enzyme, the translation initiative factor and the transcription factors. So depending on the requirement or depending on the signal, so it is going to carry out the pathway. Now यहाँ पे एक क्वेश्चन ये पूछा जा सकता है कि when does a cell understand कि उसको MAP kinase चलाना है या PI3 kinase AKT pathway करना है या JNS kinase pathway करना है. So for this you should be able to give the answer. ठीक है. आपको बताना होगा कि आप कैसे like how the cell is able to understand or differentiate like which pathway to be undertaken. So this is your assignment, guys. Okay, so let me just make the question very clear. So this is for your assignment. How does the cell undergoes or it decides which pathway, which signaling, cell signaling pathway to initiate. ठीक है. ये आपका असाइनमेंट हो गया. ये आपको सर्च करना है, आपको समझना है and be ready with the answers. So this is going to help you guys. Okay, so the critical transcription factors which are targeted by AKT are also including the members of the FOXO family. Okay, so FOXO family. So FOXO family, they get phosphorylation, right? And that creates the binding site for the cytosolic chaperones like 14-3-3 proteins. So these are very important proteins and they are going to that sequesters the FOXO in an inactive form in the cytoplasm. तो यहाँ पे क्या है 14-3-3 proteins है जो FOXO proteins को sequester करे. So once they get phosphorylated, they will be free. ठीक है. So in the absence of the growth factor stimulation, the FOXO transcription factor translocates to the nucleus and induces the जीन एक्सप्रेशन. ठीक है. तो यहाँ पे कोई है नहीं ग्रोथ फैक्टर. व्हाट इज हैपनिंग? फॉक्सो है जो अंदर जाएगा एंड इट इज गोइंग टू इंड्यूस द फॉक्सो बेस्ड ट्रांसक्रिप्शन ऑफ द जीन्स. नाउ व्हेनेवर you have got the presence of the growth factor, what will happen then? It is going to bind to the tyrosine receptors, activating your PI3 kinase. PIP2 से PIP3 हो जाएगा. AKT एक्टिवेट होगा. AKT आपका फॉक्सो को फॉस्फोराइलेट करेगा. Now once it is phosphorylated, what will happen? There will be binding sites for the 14-3-3, right? And then it will keep it in the inactive form. आपका FOXO protein को. So what will happen? Now the FOXO induced genes will not be expressed. ठीक है. So this is like in the presence of the growth factor. So another target protein is GSK. Just now we have discussed about it because it is regulating the metabolism as well as the cell proliferation and survival. ठीक है. And along with the FOXO, GSK3 is also inhibited by the AKT phosphorylation. So phosphorylation, the targets of GSK3 include several transcription factors and translation initiation factor like EIF2B. E stands for the eukaryotic. So phosphorylation So eukaryotic initiation factor 2b. Okay, this is how you have to understand. So phosphorylation of this factor, it leads to global down regulation of the translation initiation. So GSK3 provides a link between growth factor signal and the control of cellular protein synthesis. So this is how the regulation is happening. So let's understand this. So this is the regulation of the translation by the phosphorylation of EIF2 and EIF2B. So guys, अगर आप अभी ये समझ सकते हैं, so it will be going to help in understanding the future also, because in the Watson summary series I will be taking the transcription and translation. So वहाँ पे भी ये same चीज आएगा, ठीक है? तो आपको ये याद रखना होगा. So the active form of EIF2 escorts the initiator methionine tRNA to the ribosome. Yes, so initiator factor है, क्या हो रहा है यहाँ पे? आपका methionine आ रहा है, ठीक है? Now it is then released from the ribosome in the inactive GDP bound form. So whenever GDP is there, it means it is in the inactive form. But where GTP is there, it is in the active form. ठीक है? And we know that in order to continue translation, it must be reactivated by the 2B, which stimulates the exchange of the GTP for the bound GDP. तो ये कौन करके दे रहा है आपको? EIF2B, जो आपका guanine exchange factor की तरह काम कर रहा है, ठीक है? So EIF2 has got the bound initiator tRNA, and it is going and binding to the ribosome. So moment it has been released into the ribosome, the EIF2 becomes GDP. Okay, then again it will be activated with the help of the EIF2B. Now translation can be inhibited, for example under the stress condition या फिर growth factor नहीं available है. So that time what happens is the regulatory proteins will the regulatory protein kinases will phosphorylate either the EIF2 or the EIF2B. So these phosphorylation will block the exchange of the GTP for GDP, and hence what will happen? It cannot be regenerated, and वहाँ पे ही आपका translation बंद हो जाएगा, ठीक है? So guys, that's all about the today's lecture. So let us summarize. So today we talked about the PI3 kinase AKT pathway. So how exactly it is getting the activated. So whenever there is a growth factor which is going to bind to the receptor, with the help of the ancestral domain, PI3 kinase gets activated, and it actually adds the phosphate group to your PIP2 to become PIP3. So it recruits the association of the PDK1 and the mTORC2, which activates your AKT. फिर AKT आपका phosphorylation करता है, so either वो inhibitory phosphorylation होगा या activation के लिए होगा. For example, in the case of inhibitory, we talked about the FOXO protein. So जैसे ही FOXO में phosphorylation होता है, it gets bound with the 14-3-3 proteins, and hence it is not able to cross the nucleus, and where gene expression is being inhibited. ठीक है? अगर AKT phosphorylate नहीं करता है FOXO को, then FOXO induced gene expression होगा. Similarly with the case of the GSK3. So that's all for today, guys. If you like the video, please hit that like button, and if you haven't subscribed yet, please subscribe and get all the lectures related to your favorite reference books like Cooper, Watson, Leninger, etc. And if any more suggestions or any doubts are there, please you're welcome to comment, and I will be very happy to answer them. You can also write us on excelsioracademy 1228 at gmail.com. Till then, take care, guys, and keep studying.